Nathan blotting, again a technique in molecular biology which is used to study RNA. And this technique has been named Nathan because it is an analogy to southern blotting, as the technique is more or less similar. The workflow is more or less similar. Now let us look at the learning outcomes of this session. So northern blotting as a technique is based on again nucleic acid hybridization, just as southern blotting is. But here the target is not DNA, it is generally a particular RNA. So northern blotting not only identifies the RNA, it is also allowing estimating size of the RNA and also its abundance. The technique involves transfer of RNA from a gel, which is why it is called as a blotting technique, and to a nylon or nitrocellulose membrane followed by hybridization. The Nathan blot analysis can reveal expression levels of a gene, differential expression, regulation of expression, etc. So what is the workflow of Nathan blotting? So one can isolate the RNA, separate the RNA by gel electrophoresis, transfer the separated RNA bands to a membrane. This can be either a nitrocellulose membrane or nylon membrane. Hybridize the RNA with a probe, and that probe can then be detected. So, the RNA isolation per se requires the cell to be lysed and membrane to be disrupted, and then deproteination and absolutely take care of the fact that RNA is not degraded. So, therefore, RNA's inhibition is something that is addressed. So these two methods are generally taken care of by a reagent that is called as trizol, total RNA isolation reagent, and that is the most commonly used reagent that is used for RNA isolation. This contains acidic pH, guanidinium salt, and phenol, and all these three together take care of denaturing proteins and DNA, leaving the RNA behind. If the pH were alkaline, then even RNA would be separated along with the DNA. So in this case, one is taking care that only RNA is isolated. Then, of course, the phase separation is carried out by adding chloroform. So you get three different phases. The aqueous phase is what will have the RNA. And then that aqueous phase is collected and precipitated using isopropanol or chilled ethanol. So what one receives, what gets after centrifugation is a pellet of RNA. Now this RNA is separating using denaturing gel electrophoresis. Now, the denaturing gel is used because the RNA has to remain in a primary structure form and not secondary structure form. So formaldehyde can be used for denaturing. Also, RNA itself can be denatured in glyoxal or DMSO. So when it is separated, the all RNA are present in the primary structure. If it were to be present in a secondary structure or tertiary structure, then its migration can get affected, and one would not have, or one would have separation, but then one would not be able to kind of gauge the molecular size properly. And to identify the molecular size, or to approximate the molecular size, there are a wide range of RNA ladders available that is run along with the sample RNA. So by comparing it with the RNA molecular weight marker, one can find out what is the size of the DNA. Now, the next step is the transfer of RNA from the gel to the nitrocellulose membrane or to the nylon membrane. So just like in southern blotting, in northern blotting also the same principle is done, where you have the gel present on a wet filter paper, and that filter paper is dipped into buffer, generally SSC buffer, and this buffer constantly keeps the Wattman filter paper wet. On top of this gel is present the nitrocellulose membrane, and on top of the nitrocellulose membrane is present a number of dry Wattman filter papers. And then, of course, one has sponge pads added, and then to that, weight is added so that the nylon membrane is layered properly on the gel, and it is left like that so that as water moves up to the dry filter paper, you would have from the gel the DNA moving to the nylon membrane. Now we must understand that the DNA or the RNA is negatively charged, and the nylon membrane or the nitrocellular membrane has positive charge. So therefore there would be an interaction of the DNA with the nitrocellulose membrane. Now once that is done, or this is a traditional way of doing the blotting, but now what one uses is electroblot. So the same stack of gel, nitrocellulose membrane, and papers are now introduced in a transfer chamber with two electrodes, a negative electrode and a positive electrode. And when the current or when the pulse is given, then you have current flow from the negative electrode to the positive electrode, which is how the DNA from the gel moves to the nitrocellulose membrane. So the transfer in this case is faster than the transfer in this case. Here it would take hours to do so, while here comparatively it would take lesser time. Then, of course, once the DNA is present on the nylon membrane or the nitrocellulose membrane, then it has to be cross-linked. So if it is a nylon membrane, it can be cross-linked by exposing it to UV radiation, while if it is a nitrocellulose membrane, then the nitrocellulose membrane can be baked at 80 degrees for a stipulated time. Generally, it is 2 hours. So that way what happens is that the sugar phosphate, which has negative charge, is now firmly attached to the positive charge of the nylon membrane. So we are basically ensuring that the RNA is tightly associated with the membrane. Now one needs to carry out hybridization and detection. So first, pre-hybridization or blocking is done. So the nitrocellulose membrane containing the RNA is treated with a blocking agent. So the blocking agent will go and bind to all those areas where the RNA is not attached. This is basically to prevent spurious binding of probes to the nitrocellulose membrane. This kind of ensures that the probe only goes and binds to the RNA present on the nitrocellulose membrane. So the next step is hybridization. So probes are added, or the nitrocellulose membrane is put in a solution containing the probes. So the probes will bind specifically to RNA that has complementarity, and then it is detected. So only those RNA will be observed after detection which has complementarity to the probe. So that is how an RNA can be identified from a mixture of RNAs. So this is another hybridization membrane after hybridization and detection, and as you can see very clearly the RNA is visible. So, what is the analysis that can be done from here? One, the RNA, specific RNA that one wants to identify can be detected. Secondly, what is possible is that the size can be identified. So one can match it with the RNA molecular weight marker and kind of look at what is the size of the RNA. One can also see whether it is differentially expressed or not. In this case, one can very clearly see that this RNA has very low expression rate in normal cells while it has high expression rate in the cancer. So it is differentially expressed. Sometimes you may not even find the RNA expressed in one cell type, but it is the DNA expressed in one cell type, but in another cell type it is getting expressed. So differential expression can be looked at by looking at whether the RNA is present in a tissue type or not. And of course, the level of expression, the intensity of the bands itself is saying whether the expression is high or whether the expression is low. So one can see that epidermal growth factor RNA is high in the cancer cell, but it's very less in the normal cells. So these are the analysis that can be done. So expression studies is something that is enabled by northern hybridization. So let us make the conclusions. RNA is isolated from a particular cell type or types and then separated using denaturing agarose gel electrophoresis. The separated RNA are transferred to a nylon or nitrocellulose membrane and treated such that the single-stranded RNA is cross-linked to the membrane. The membrane is subjected to pre-hybridization treatment to ensure that there is no non-specific binding, and then it is followed by hybridization with a specific probe. The northern plot post-detection can be analyzed for size of the RNA, differential expression, and also the level of expression itself. So, with East Southern developing a technique to identify DNA and naming it Southern blot technique, the technique was extended and modified to analyze RNA, which therefore being poles apart, pun intended. So, if you have DNA, it is Southern, but if it is RNA, it is Northern, and hence was named as Northern blotting. Thank you.